An organism's genome is all of its genetic material. So all of its DNA, all of its RNA, no matter how it's packaged within the cell. And when we look at an organism's genome, we can see a few different things, and especially if we're looking at humans or eukaryotic organisms versus our bacteria and archaea, our prokaryotic organisms. So all of the cell's activities come from the information that's found in DNA. And so the sequence of those bases, the A's, the T's, the G's, and the C's, represent the genetic information. And so each piece, each little segment is called a gene, like we referred to. The full collection of genes is referred to as an organism's genotype. But the key thing to keep in mind is that an organism may not express all of its genes or all of its genotype at the same time. Certain genes can be turned on or expressed or turned off as necessary. The set of genes that are turned on at any given time and that provide for an organism's characteristics that we can observe is called its phenotype. So the genotype refers to its actual genes, its genetic makeup, that sequence of A's, T's, G's, and C's. And then the phenotype is what it looks like. So if we're talking about humans, the phenotype would be the brown hair and the green eyes and the dark skin and the height and the freckles and the way the nose is shaped or the mouth is shaped. So the phenotype is that physical appearance. In terms of bacteria, the genotype is not going to change. It's going to remain constant, but the phenotype can change depending on conditions in the environment or nutrient availability or whatever. And so an example of that is Seratia Marcescens. This is a pathogen. We at once thought that it was not pathogenic, but we now realize that it's a pathogenic bacterium, and when it is grown at 28 degrees, it produces a red pigment. When it's grown at 37 degrees, it doesn't. And so this would be an example of how its phenotype could change based on environmental conditions, even though its genotype stays the same. In the right situations, in other words, a temperature at 28 degrees Celsius, the gene that codes for a red pigment is turned on. And so we get the red pigment. But at 37 degrees, that gene is turned off, and we don't get the red pigment. Now, most of an organism's genome is organized into its chromosomes. Remember, we discovered chromosomes before we realized their significance. Eukaryotic chromosomes are found in the nucleus. Prokaryotic chromosomes, typically it's a singular circular chromosome found in the area that we call the nucleoid. And depending on the organism, we can have, thousands of genes. And the more complex an organism is, typically the more genes that it's going to have. In eukaryotes, the chromosomes are usually diploid, meaning that we have 2 copies of each one. But in prokaryotes, they're typically haploid, meaning we only have 1 copy of each chromosome. And in addition to our genes, so here's our chromosome. If we unwind that and we look at our genes, we're going to have areas in between the genes that is called non-coding DNA. And this used to be referred to as junk DNA. But it was renamed, or we've kind of got rid of that nickname, because we do realize that even though these areas of the DNA do not code for a protein, they may have valuable information on them. We don't really understand all of the roles of the non-coding regions of DNA, but for the most part, we think, yeah, a lot of it doesn't have purposes within the cell, which is how it got that nickname junk DNA in the first place. Now, most DNA is found within the chromosomes, but some cells have other pieces of DNA outside the chromosome, which we call extrachromosomal DNA. Now, it's still part of its genome because remember, a genome is all of its genetic information, but it's not found within its chromosome. In eukaryotic cells, this would be chromosomes from like mitochondria or chloroplasts, because we know that we have mitochondrial DNA and chloroplast DNA. But for prokaryotes, they have loops of DNA referred to as plasmids. And plasmids can contain one or more genes that are not necessarily for the cell to grow, but contains information that could be beneficial for survival or growth under special conditions. It could lead to an organism being pathogenic, so it could code for some virulence factors. And we know that bacteria can swap these plasmids. They can transfer these plasmids back and forth, and that's called horizontal gene transfer. You've got a little case study here in this section that talks about Bacillus cereus and Bacillus anthracis. And Bacillus cereus and Bacillus anthracis are very, very closely related to one another. In fact, they believe they may actually be the same species. But Bacillus anthracis contains two plasmids that code for the anthrax toxin, and that's what makes it lethal. And Bacillus cereus doesn't cause infection at all because it doesn't have that information. So the pathogenicity of Bacillus anthracis, or its ability to be a pathogen, to cause disease, is due to the information that's contained on its plasmid. So prokaryotes are a little bit unique in this respect, that they have these plasmids that can, give them extra information.